You see the slides now?
Yes.
Okay, cool. Um, yeah, so thanks for the invitation to speak today. And the work I'm talking about today involves a lot of people, and so I just want to just sort of visually show you who some of these people are. It's a very long list. If I put up a list, the text would be so small that you wouldn't be able to really appreciate all the contributions of all the people. Um, and I also, want to thank my family because, uh, ever since I started doing this, my family has been coming with me and giving up their summers to do field work, and they get to have a little fun in the process. But um, you know, uh, I appreciate their love and understanding over the years in terms of making this work possible. The talk today is going to be a little different than probably most scientific talks you've seen. Um, Typically, science talks focus on what? This is what I did. Um, Sometimes they'll talk about the how, they'll talk about methods, but when I talk about how, I'm talking about a bigger how than just the materials and methods and whatnot. Um, but very, very rarely will you ever hear anyone talk about why. Um, they may give you something in their introduction about, why this species is important, why the management implications of, a particular fisheries need to be studied, blah, blah, blah. But again, I'm going to talk about a bigger why. And I'm going to start the talk by offering a unusual statement for a speaker, which is, I should not be here. And the reason I say that is that I'm a Mexican American evolutionary biologist and marine scientist. In 1997, the year that I completed my PhD, Latinos and Latinas only accounted for 3.4% of all the PhDs earned in biology. Um, for marine science, that same year, it was probably one or two. I can say one or two because the numbers are so low. The data that you can get from NSF is aggregate data. And so for Earth, planetary, and ocean sciences, there are 15 Latinos or Latinas that got their PhDs in 1997. And only about 10% of that larger group are actually ocean science PhDs. And so somewhere between one and two, you know, got their PhDs then. But I would say there's about a 0% chance that I would have become a marine scientist and a professor. And it's not necessarily because of strong declarative statements that I made as a graduate student, such as I never want to be a faculty member at a research intensive university like Berkeley or UCLA. Um, that is me in 1996, around the time when I said that. Um, but instead, the reason why it's such a low probability that I would end up on this path is that I was born in Tucson, Arizona. For those of you unfamiliar with Tucson, Tucson is a desert. It is a long way away from the ocean. Um, I did grow up surrounded by animals, but the important thing to note in this picture, other than it's the only image in my entire talk where you'll see me wearing diapers, is that uh, these rabbits are not pets. These rabbits are food. My parents were raising these to sell to the butcher to make a little extra money because in the household that we came from, money was scarce. I went to low performing schools. This is my high school, Tucson High. Tucson High was not visited by college recruiters. We were visited by military recruiters. Um, and, that's because, less than 40% of the students I started high school with as a first year actually continued on to graduation. Um, I attended the University of Arizona. That was not my aspiration. I wanted to go somewhere else, but that is ultimately what I could afford, my hometown university. And I wasn't sure what I wanted to do um, career-wise. One of my big goals was becoming a professional classical musician. I'm a classically trained percussionist. I would have loved to have had a job, playing in a professional symphony orchestra. But I also really liked fish. Um, and so I studied music and biology at the University of Arizona. Um, and it was only at the end of my junior year that I took these two classes from Marilyn Hauk and Chuck Lowe. Um, Marilyn taught an animal behavior class, Chuck taught a herpetology class. And for both of their classes, they required that we did some kind of small research project. Um, I never really thought about doing research, had never done undergraduate research up to that point. But I figured, given that I was spending, 20 hours a week in the practice room playing percussion, I would be efficient and I would study the behavior of a frog. Um, this frog, particularly the canyon tree frog, Hyla renicolor, and the behavior I was interested in is that that frog looks a whole lot like a piece of granite. Um, but that only does you any good from an evolutionary perspective if you're hanging out on granite. If you hang out on a leaf, you stick out like a sore thumb. And so I wanted to know, like, could the frogs tell the difference between being on a substrate where they blend in and where they don't? Um, it was not a very good project at all. It didn't work very well. But um, at the end of that semester, both Marilyn and Chuck independently said to me, it's like, you know what? You should go to graduate school and you should go to Berkeley. And so I did. I applied to graduate school. I went to UC Berkeley to the Department of Integrative Biology. And um, I didn't really know why I was going to grad school other than it got me out of Tucson, which was, my primary goal in going to graduate school. And I went to graduate school to continue studying this cool little tree frog. And what I thought was really cool about this frog is that it lives in these tiny little pockets of aquatic habitat that are surrounded by a sea of desert. So you can see this little pool here. It's surrounded by lots and lots of desert. If you zoom out, what you will see is what is known as a sky island. These sky islands themselves are these islands of habitat that rise up out of this vast expanse of desert. And you can actually see this from satellite images. So all of these little green patches that you see, I don't know why my computer did that so quickly, but all those little green patches you can see are sky islands. And within those sky islands are these little tiny islands of habitat. And I can't tell you why I wanted to know this, but I wanted to know how'd the frogs get there? And um, my goal was to use genetics. PCR was very new, DNA sequencing was very new, and I thought, I can use, mitochondrial phylogeography to reconstruct the evolutionary history of these frogs, figure out how they're related, and maybe that would give me an idea of how they got there. I spent my first 2 years of grad school trying to get the lab methods to work. It was an abject failure. And so after 2 years, I decided, you know what, I got to do something else. And I got interested in this animal here, Mungos mungo, the banded mongoose. And I was interested in these because they have all of these eusocial behaviors that suggest that they may be more genetically related to each other in their family groups than you might expect, for a normal outbreeding population, kind of like, honeybees or eusocial insects. They're more closely related to each other. And that's, you know, how those eusocial behaviors essentially get selected for over time. Um, in the process of developing this project, um, I got sucked into a different project, um, which was studying the maternal behavior of spotted hyenas. Uh, Hyenas are really interesting in that it is a matriarchy in captivity. Female hyenas typically give birth to twins. The sex ratio is equal, but in the wild, what you see is that high-ranking females have a disproportionate number of male offspring. Low-ranking females have a disproportionate number of female offspring. I know we're talking about hyenas in a talk focused on fisheries. Uh, we'll get there. Um, but anyway, I joined this project. This was going to be my PhD thesis. I went out, spent time out in the Masai Mara, radio collaring hyenas and following them around and studying them, living in a tent on the side of a river by myself for a very long time. But uh, it very quickly became apparent that this project was not going to work. And so yet again, I had a failed PhD project, came back to Berkeley, and then with some fellow graduate students of mine, this is not Chris and Alan in grad school, um, this is later on working together in Indonesia, but Chris and Alan and we put together a proposal to use genetics to look at larval dispersal of things out in Indonesia. I was going to look at clownfish. Chris was going to look at cowries. Alan was going to look at sponges. We put a lot of time into developing this, and then it didn't get funded. And that brought me to the end of my fourth year of graduate school when my PhD advisor decided to leave for another institution. So I entered my fifth year of grad school with no advisor, no project. Um, and fortunately, um, a new faculty member by the name of Tyrone Hayes, who um, Tyrone and I were actually graduate students together at Berkeley. Um, he was 4 years into he graduated with his PhD in 4 years, was 6 months into a postdoc, and Berkeley hired them back to their faculty. I think he had tenure like 3 years later. He's unusual. That's not the typical trajectory for people who are interested. But the cool thing about joining his lab was that in the 2 years since I had originally abandoned my frog project, the technology surrounding DNA sequencing changed significantly. And what was previously impossible became possible. And I actually circled back to my original PhD project and got genetic data from all of these populations. And the cool thing is that there are these different groups, and these different groups are actually, why are something is very glitchy with PowerPoint on my end, but all of these groups actually are united by the watersheds that they border. So essentially, these frogs got to these isolated locations now back in the Pleistocene when things were wetter and cooler, and there were probably these populations spread throughout this region along the watersheds. And then as we entered the Holocene and things began to dry up and become desertified, these little relic populations remained in these areas that they used to be. So, um, with that, I was planning on a career in herpetology of some sort. When in the fall of 1997, as I was finishing my PhD, this paper came out by Callum Roberts. And Callum Roberts was interested in figuring out how do you set up networks of marine protected areas in ways that maintain ecological function and connectivity. And so he came up with this idea that if you look at an area like the Caribbean and you know um, what the patterns of the oceanographic patterns are, all you need to do is to look at how long fish larvae develop in the water column, and you can predict exactly where they'll go. So if a fish larva is in the water column for 1 month, on those ocean currents, it may only go a relatively short distance. But, if it's out there for 2 months, it may go much, much further, not just because it's in the water column for a longer time, but it enters part of the Gulf Stream where the currents are and the transport is much faster. And when I read this paper, the thing that immediately struck me about it as being so cool is that I happen to have a data set that showed that it was wrong. And the reason I had this data set is that um, one of my fellow graduate students at Berkeley, Mark Erdman, he had spent his entire PhD thesis studying mantis shrimp or stomatopods in Indonesia. He was looking at stomatopods as bioindicators of anthropogenic stressors on coral reef ecosystems. And wherever he went, when he was collecting his stomatopods for his thesis, he saved those and preserved them in ethanol. And the reason I got involved was that he basically discovered what he thought were like 13 new species of stomatopod, but he wasn't really a stomatopod taxonomist. He wanted to have some independent evidence to suggest that, yes, these things were really different. And so he engaged me in this little side project looking at the molecular systematics of these stomatopods um, in Indonesia. But as we did this and we were looking at interspecific variation, we also looked at intraspecific variation. And one of the things that we saw, just coincidentally, because he provided me with with four population samples from one species in Indonesia, and they happened to be in northern Sulawesi and then down in Java and in Bali. And it's essentially at the mouth of the Sunda Strait and the Indonesian through flow, which is a movement of 20 million cubic meters, can't talk today, 20 million cubic meters of water per second, flows through the Makassar Strait there. And so, in this animation, which is not looping like it should, what you see is a lot of water passing through this area, which should essentially dump larvae from these northern Sulawesi populations down into the bottom of the strait, creating high larval dispersal and high gene flow. But genetically, these populations were very, very different. Um, and so from that, I took that preliminary data and wrote a NSF postdoctoral fellowship proposal, which got funded, and took me to Harvard to study this phenomenon and use genetics to track larval dispersal in marine ecosystems. We looked at multiple species, and I'll just show you data from one. But what we found was that populations from southern Indonesia here look very different from populations in northern Indonesia. And uh, the reason why we think that this happens is that if you see here this light blue area, this is all the Sunda Shelf. And what happens when glaciers are building up on land and creating that cooler, wetter habitat that allowed my frogs to disperse throughout southern Arizona. Why is this not playing? All right, something is very glitchy today. Anyway, I'm going to just flip through this. As those glaciers build up, sea levels drop, and what you see is that area of the Sunda Shelf becomes terrestrial environments. And you have essentially a strong constriction of water flow from the Pacific to the Indian Ocean. And, you know, the glacial cycles were intermittent, but they lasted for quite a long time. And they lasted long enough, what we think, to essentially drive genetic isolation between populations on the Pacific side of the Indonesian archipelago and populations on the Indian Ocean side of the archipelago. So this study essentially helped propel me to my first faculty position at Boston University as part of the Boston University Marine Program down in Woods Hole. Ultimately, Boston University decided that they weren't going to support a marine program down in Woods Hole anymore because they didn't see the value of having a marine program in Woods Hole. Um, Not sure about the logic there, but uh, that's what happened. Um, and during my time at Boston University, I continued to work in Indonesia and started to develop a research program focused on this region called the Coral Triangle. Coral Triangle is the world's largest and most biologically diverse marine ecosystem, 6 million square kilometers spanning 6 countries. If you look at global biodiversity patterns, what you see is that, the Coral Triangle here has the highest marine biodiversity of anywhere in the world. And whether you go north, south, east, west, away from the Coral Triangle, there is a very sharp decline in marine biodiversity. And I became interested in how this became the global epicenter of marine biodiversity. And at the time, there were a couple of competing hypotheses. One, in fact, at the time, the leading hypothesis was that this was a center of accumulation and that speciation occurred in the isolated habitats of the Pacific and the Indian Ocean, and that over time, those species expanded their ranges into the Coral Triangle, um, generating the elevated levels of marine biodiversity that we see. Another hypothesis was that this region was actually a center of origin and that speciation occurred within the Coral Triangle. And rather than accumulating marine biodiversity, it's actually exporting this novel biodiversity into these peripheral islands of the Pacific and the Indian Ocean. The problem with this hypothesis was that there is no mechanism to explain how this would happen because, the thought was, and largely still is, that speciation requires isolation. It requires some sort of physical separation that leads to genetic differentiation to the point that populations are no longer reproductively compatible. How did that work in the ocean? It's like all of these things that are living in the ocean, most of them have some sort of larval dispersal period. Those larvae should travel on ocean currents. That should facilitate gene flow and, you know, uh, essentially facilitate panmixia. And there shouldn't be any opportunities for speciation within the coral triangle itself. Um, but what we did is we went out and we studied lots of different species, reef fish, pelagic fish, snails, clams, sea stars, stomatopods, and whatnot. And what we showed is that just like that original stomatopod I showed you, lots of species had this signal of genetic divergence between Indian Ocean and Pacific Ocean populations. The other thing that we found was that in a lot of species, particularly clownfish, giant clams, stomatopods, there's also a strong genetic break across a region called the Maluku Sea. And so here's some data from one of the stomatopods that shows this pattern. And the Maluku Sea is this area of ocean between the islands of Sulawesi and Western Papua. And this is where it's important to acknowledge that as someone who studied herpetology and grew up in the desert and went to a school that had no marine classes whatsoever, I have no formal training in marine science whatsoever. And so I actually had no idea there are these things called the Western boundary currents. And again, my animation is not looping like it should, but what you should see if this was working is essentially water moving along the northern coast of the island of Papua till it hits this island called Halmahera. At Halmahera, those waters are retroflected back to the east on the north equatorial countercurrent, creating this feature called the Halmahera Eddy. This oceanographic process limits the transport of water from Western Papua and indeed the western part of the Coral Triangle across the Maluku Sea. And because the water isn't making it across the Maluku Sea, the larvae that are in the water don't make it across the Maluku Sea. And so, had I studied oceanography at all, I would have actually started by predicting that we would have seen a genetic barrier here. But, needless to say, this is probably why we're seeing this strong genetic break at the Maluku Sea in lots of different species. Now, to look at this, we looked at a lot of different species, saw that pattern, but we also worked with some oceanographic modelers, so Jonathan Kuhl and Eric Tremmel, and they create these coupled biophysical models where you can essentially create a virtual ocean that has virtual populations on virtual coral reefs. You can then start releasing virtual larvae into this virtual ocean and see where those larvae go. And, on a generation by generation basis, what you see is that, this diagonal here, the reason that you can see that as a line, there actually isn't a line there. It's just a bunch of data points that show that most of the larvae in the simulations don't actually go anywhere. So on average, a larvae from Sumatra ends up in Sumatra, one from Makassar ends up in Makassar. But every so often, a larva from Makassar ends up somewhere else, like down in Flores. And you actually have dispersal and connectivity. And the cool thing is that because this is virtual larvae on virtual reefs in a virtual sea, you can run this experiment over and over and over for thousands of generations and then summarize what connectivity patterns um, result. And what you see is, red here is high connectivity, blue is low connectivity. And what you see is that there's limited connectivity across the Maluku Sea, and there is limited connectivity across the Sunda Shelf region. So um, this is absolutely consistent with the genetic data that we see, where central Indonesia populations have high connectivity, and then there are these distinct populations to the west, or to the east and to the west. So, going back to this, original question of how the Coral Triangle became a global epicenter of marine biodiversity, what our data suggests is both because of historical processes and contemporary oceanography, there are strong limits to larval dispersal, creating barriers to gene flow, allowing diversification to happen, and speciation to occur. Now, from an evolutionary standpoint, that's interesting. That helps us understand, this biogeographic pattern that we see. But the important thing, at least for me, from the perspective of a conservation biologist, is that this data was really useful in helping people think about how to set up marine protected areas in Indonesia. And in particular, the data that came out of this project over, more than a decade resulted in more protection for populations in eastern Indonesia, in the region of West Papua, but also in Western Indonesia, in Java and Sumatra, because those populations are incredibly unique genetically, even though they fall outside of the Coral Triangle. So we spent a lot of time in Indonesia. We spent about 20 years working out there. Um, but we've been doing stuff more locally as well. Um, we've been doing a lot of environmental DNA work, eDNA. Uh, I think at this point, most people probably know what it is, but I'll just say briefly. So you can essentially go out, drop a Niskin bottle, collect a bunch of water. In that water, there is DNA. You can isolate that DNA, bring it back to the lab, and sequence it. And from those sequences, use DNA metabarcoding to essentially reconstruct, what species were in that area that you sampled. And so it's a really powerful tool to be able to go out into these ecosystems and understand community composition, even when, a lot of the things that are in that community you may never actually see on a dive. I've done, dozens and dozens of dives in local kelp forests here. Uh, I've seen giant kelp bass or giant black sea bass. I've seen them 3 times. Most of the times you don't see them, but eDNA picks them up. The eDNA can tell you that they're there. We just don't see them most of the time. But um, one of the cool applications of eDNA is that you can use it to look at the effectiveness of a marine protected area. So one of my former students, Zach Gold, who now heads up marine genomics lab for NOAA, um, he used eDNA to look inside and outside of marine protected areas to see if, instead of putting divers in the water to show that your marine protected areas are having an impact, could you just use eDNA? The answer is yes, you can. You can differentiate the communities from MPAs, from outside of the MPAs, and samples that you take at the edge of the MPA look like a mix of the MPA and outside of the MPA. Um, Another cool thing that he did was he used preserved larval samples from the Cal Coffee longitudinal sampling program. They've been sampling fish larvae in the same places year after year since the 1940s. Fortunately, most of those have been preserved in ethanol. You can extract the DNA from the ethanol, and then what you can do is you can um, look at those samples over time. Here we've got year. This goes back to 1995 to 2000. Sorry about that. Um, and there's this big marine heat wave that occurred in 2016. And what you see associated with that heat wave in the DNA that we get out of those bulk samples of fish larvae is that cold adapted species like the northern lampfish decrease substantially in abundance, whereas warm water species like the Mexican lampfish increase substantially in abundance. And so without going into the larvae, without having to, you know, identify these tiny little larvae, which many times you can't through morphological analysis, you can just use genetics to show that these populations are changing over time, which is super, super cool. Um, Other things that we're doing now, we're using full genomes to look at differentiation of marine fisheries targets in California, rockfish, California market squid, spiny lobster, Dungeness crab, and California halibut, and looking for stock structure, but also things like adaptive genetic variation to see uh, whether they have the ability to adapt to climate change and whether that variation can disperse quickly enough through natural processes to get to the areas as habitat as environments are changing. So this is a little bit of the what we're doing and what we've done over the years. Um, but I wanted to return to where we started, where, this is work that's being done by, a kid that grew up in the desert, went to shitty schools, studied frogs, and somehow became a marine scientist. Because as I was making that transition from studying frogs to being a marine scientist, and I started to go to scientific conferences, um, I started to notice that there was not a lot of diversity in the room. So the United States, about 33% are people of color. In STEM, that drops to 11%. In marine science, it drops to 4%. And this was really, really apparent to me when I first started going to conferences. And so, what I wanted to do um, was to create a program, which I did, called the Diversity Project, that focused on expanding opportunities for people in marine science, students from schools where, you know, like me at Arizona, we didn't have the opportunity to study marine science. People who, grew up in inland parts of the country like I did, that didn't necessarily have access to the coast. And so um, every year since 2005, wow, this is like, not sure what is up with my computer today. Something not good. But um, we've been recruiting and taking students um, out to places like Indonesia or French Polynesia, training them to be scuba divers, training them to use scuba as a research tool, and then mentoring them through the process of developing their own research projects. And um, over the years, we have had 120 alumni 20 of them have already earned PhDs at places like Harvard, Stanford, UCLA. There are 31 students currently in PhD programs. 19 students have earned master's degrees, 3 Fulbright fellows, 18 recipients of the NSF GRFP, 6 tenure track faculty now are alumni from our program, 2 staff scientists at the Nature Conservancy, 1 at the World Wildlife Fund for Nature, 2 members of the uh, of 2 members of the Association for the Study of Limnology and Oceanography Society, and so all of this work that I've talked about doing in Indonesia, all of this was done in the context of the Diversity Project. And so, because of my experience, what I decided to do was like, I wanted to do the science, but I wanted to do it in a way that created opportunities for others. And, um, people like me who never really had the opportunity to consider marine science as a, a career choice, as an undergraduate. So, um, while we were doing the diversity project in Indonesia, I became increasingly aware and concerned. Are we frozen here? Um, about this pattern here, where the red and yellow indicate coral reefs that are either threatened or severely threatened. Only the blue ones are considered healthy. And, working in the Coral Triangle, 80% of the reefs in Indonesia and the Philippines are threatened or highly threatened. And um, we figured, if we're doing this program to create opportunities for US students, maybe we should be expanding this to create opportunities for Indonesian students as well. And so we created what was known then as the Indonesian Biodiversity Research Center. We created a team of people from a variety of different institutions, both in Indonesia and in the US, to essentially train and inspire a new generation of marine scientists. And we developed short courses to teach students how to do marine biodiversity research, um, using both traditional methods that involve taxonomy and identifying and counting things, but also using modern genetic tools. And over the time that we were working in Indonesia with this IBRC, we built 2 labs and built a field station. We trained more than 300 Indonesian students. This resulted in dozens of papers, more than $11 million of grant support to Indonesian and US scientists. But like to me, more than the numbers are the people. And, it's Martha, who's, a faculty member at Yale. It's Alexis, who heads up the Pacific Fisheries Program at TNC. Um, Melissa Kemp, who's professor at UT Austin. It's people like Camille and Tiara who formed an organization called Black in Marine Science that is having a major impact in diversifying marine science. It's people like Aaliyah who created a nonprofit called Mahogany Mermaids, trying to get young Black girls interested in scuba diving so that they will consider careers in marine science. It's people like Rita, who work now at the Ministry of Fisheries um, in Indonesia, Dita, who's a professor at Diponegoro University, Aji, who works for the Nature Conservancy's Blue Carbon project. These Indonesian scientists that are having a major impact on the science and conservation of those marine ecosystems. And I want to highlight one particular person, Andre, because he developed a project while working with us where they went out to places where they essentially were selling shark fins wholesale into the shark fin trade. By getting samples of those and using DNA barcoding, they basically showed that reef sharks have largely been extirpated from most places in Indonesia, and that fishermen were targeting CITES red listed species like scalloped hammerhead sharks. The Indonesian government didn't have any legislation protecting them because the Indonesian government didn't know that there was a fishery. So this is the first time we demonstrated that there's a fishery, and that resulted in new legislation protecting those sharks. Um, and Ultimately, the IBRC transitioned into a new Indonesian-run nonprofit called Bionesia, run by Andre and a lot of the alumni of the program that we ran at the Indonesian Biodiversity Research Center. And so, um, it's the impact of thinking not just about the science, but how to do the science and do it in a way that creates a more access to more people. Um, to me, this is where I think more scientists should be working like this. So to wrap things up, I'm going to start where we started. I started by saying I should not be here. Um, you know, Mexican American kid from the desert, shitty schools, studied frogs. There's no reason that I should have become a marine scientist. But that is me focusing on probabilities. And as scientists, we spend a lot of time focused on probabilities and probably not as much time thinking about possibilities. What is possible? What can we do? We think about collecting our data and advancing our projects and publishing our papers. And yes, that's important, but what I would suggest is that if instead of focusing on the data, we focused on the people, and sorry, the computer's going crazy, we focused on the people and focused on creating opportunities for everyone to participate in this enterprise. And, this is where we're going to have the biggest impact with our science. And I think more than ever, we really need to think not just about the science that we're doing, but think about how we're doing the science so that we can really maximize the impact. So, thank you for listening, and I'll be happy to take any questions.
All right, that was absolutely amazing. Just as good as I remembered, if not better. So again, thank you so much for taking the time to come speak with us. I like brought in my daughter because she has the day off from school to come watch your talk. So we do have some time for questions. I don't know if you can stay a little after one, Paul, but
Yep.
okay, awesome. So if anybody has any questions they have for Paul, make sure you raise your hand, so that way we're not all like bombarding Paul at once. Brett, I
I've
see you
got a,
taking yourself off mute, so I'm just
got
gonna
a
go
hard
ahead and
question.
call on you.
I've got a really hard question. First of all, that was a really fascinating, fantastic talk. I mean, this is the inaugural lecture in this series. Thanks for putting the series together. So I didn't see too many people. I feel like this would be such a helpful talk for so many people. I saw you recorded it, so hopefully I'm glad your daughter got to watch it. But the hard question is, and maybe you don't want to answer, which is fine. But in the current political climate, this talk seems in particularly timely, and I'm just wondering what can be done. What have you thought about what you can do, or how the current political climate may impact these programs that you've contributed so much to?
Um, well, of fortunately or unfortunately, I think probably more fortunately right now, um, you know, the last federal grant that I have that supports this program, I filed the final project report on Thursday evening. Um, and so at this point, this is not funded by the federal government. Um, but the other thing to highlight is that um, you know, this isn't something where we're saying that, you know, unless you're, unless you're black, unless you're Latina or whatever, like you can't participate, you can't apply. I mean, we take this notion of creating opportunities for others, you know, like very seriously and interpret it quite broadly. And as I said, you know, there are other people that grew up in the desert or grew up in the Ozarks, that went to schools that don't have marine science programs. There are students at community colleges that don't have that sort of exposure to research, and so I think that um, one of the ways forward is really thinking about how, how we're trying to reach out and serve people. And, I think if you go to the community colleges, if you go to tribal colleges or HBCUs, if you recruit from, you know, universities that don't necessarily have marine programs, um, you're gonna end up with a very uh, diverse group of participants. Um, you know, and I mean that in the broadest of terms. And so you know, um, I think that you know what people need to, you know, try to avoid is, making themselves easy targets by, um, you know, selecting in ways that, you know, um, are going to get your funding cut off or get your university sued or, um, so, I think there are ways to do this, that are as inclusive as can be. And, you know, it's just a matter of, thinking creatively on how to do that.
Right.
Thanks. It's almost, yeah, it's almost like saying you can use this as an opportunity to make your programs even more inclusive and diverse.
Yep.
Thanks.
Yeah, thanks for speaking on that. Jessica, I saw you had your hand up a bit. I don't know if you still have a question or
Yeah, it was similar to Brett's question. And I just wanted to say, Paul, thank you so much. I loved your story. I went to community college for like 8 years because I was like, I don't know what I'm doing. And I feel like a failure. And then it was really hard in grad school and the projects failed 1,000 times. And I just enjoyed hearing your story and everything you do. So thank you so much.
Thanks.
okay. Kim and then Matt.
I'm muted. I'm unmuted now. I just kind of wanted to echo what Paul was saying about like the importance of like going out to community colleges and like what Jess said too. Like my community college had no ecology, it had no environmental science, and I knew I wanted to go into that. But like I didn't even have any sort of opportunity. I had to go find my own opportunities to be competitive for, going to a four-year school. And and I didn't even think that I was ever gonna even be able to go to grad school because I thought like how am I ever gonna afford it? And it was just one of my professors at community college was like, well, if you do research, like you can get things funded. So just like having people even just like putting that out there, having like being able to visit, community colleges and letting them know that those options exist. Because if you don't grow up, like my parents didn't go to college, no one around me I knew went to grad school, like you don't even know that that's possible for you. Um, and like now that I'm in my position I'm like trying to let everybody know about those opportunities because they're not necessarily hidden, but I think it is really important to connect with those, with schools outside of, and I know it's hard with UCs to do like internships with outside schools, but even just having volunteers for field work or giving a lecture can really just change things for people.
Yeah, it's. I mean, I would say that it's if you have connections with community colleges, like at UCLA, we have programs where, you know, grad student led programs where they're recruiting students from community college to, participate over the summer in research. And, um, there's a lot of cool things um, that people are doing. And a lot of the coolest stuff, tends to be, student led. And so, I guess two things I'll say is like, if this is something that, if this is an area that you're passionate about, I wouldn't wait until you're a faculty member to do it. Um, what I would say is that, as you engage in this work and engage with students in community colleges or tribal colleges and, work on creating these opportunities, do it in a way where it's a win-win. Um, a lot of people, the mistake they make is they try to make it they don't necessarily try to make it extra, but they develop things in a way where they've got their research and then they've got their other stuff. And you know, time is a zero-sum game. Um, every hour you put into one thing is an hour that you can't put into something else. And so I think one of the things that's been really essential for me, looking back on my career, and I did not do this, this was not a strategy. It's just sort of how I develop things. And looking back, I can see how important it was. But you know, my teaching, my outreach, and my research are all completely integrated. So, you know, all of my outreach advances my research. All of my research advances my outreach. You know, I publish papers from classes that I teach. And so my teaching is advancing my research, and my research is advancing my teaching. And so the more that you can think strategically about how to develop, anything that you want to do that you find meaningful, to, in your case, uh, make things more accessible to community college students. Figure out a way to do that it's a win-win for both of you, so that um, you're advancing your research and your career at the same time you're creating those opportunities.
I love that. That's amazing. Matt,
Hey, Paul. Thank you for that what everyone else has said. I mean, that was great. I think what you just mentioned spoke to some of my question, and I also appreciate you taking the time to stay uh, on for another few minutes.
Yep,
But
no problem.
I was wondering your thoughts about the role of professional societies like AFS, who are not strictly academic and who straddle a whole bunch of realms within a discipline, right? So we have our membership largely aren't academics. There's a large student membership, but it's mostly agency and private professionals who work for state, federal, and local groups and private enterprise. And many of us participate because we feel that these sorts of professional societies provide an outlet to do the type of work that you describe that may be sort of a little bit separate from what our day jobs might be. And I guess I give that was mostly context. And my question is, as a society, as a professional society, what do you see as the best way for us to engage with programs like yours? Is it, is communication and facilitating, spreading the word that these programs exist the most effective? Is it like a dollars and cents kind of contribution? Is it partnerships where, other types of partnerships? Can you speak to that a little bit?
Yeah, so thanks for the question. I think particularly when you start talking about people who aren't academics and, it's more, you know, management agencies, industry, etc. Um, I think one of the things that you guys can do that would be really valuable is thinking about, programs like, like our program, like we're not the only one, there's other programs that are out there, but looking at them as conduits where, you know, we're doing that initial stage of like training the students, developing them as early scientists, giving them exposure to these tools. Um, but they need continued development. They need continued exposure. And, one of the things that, like a lot of our students, are often looking for, after they've done our program is like, okay, like I'd like to do something that, helps me build on my skill set, do something that, maybe gets me a little closer to the conservation and management world, something that maybe gets me, closer to, regulatory agencies like U.S. Fish and Wildlife or NOAA or NIMS or things like that. And so I think, creating, um, if you have opportunities for internships and, can get the word out to the programs that are doing these this sort of foundational training, it would be great to have a place for our students to go for that next stage of their development. And you know, so if that sort of thing is a possibility, that would be awesome. And, if you want to talk more offline about it, I'd be happy to chat.
Thanks, that's helpful. And I might reach out. I think that's something that, full disclosure, I'm a federal employee myself, and so I think that that's top of mind in a lot of ways right now.
Yep.
Awesome. Are there any more questions for Paul? All right. Well, thank you so much, Paul, for joining us. There were actually a lot more people signed up, so I think people forgot they signed up. We had like almost triple the amount of people signed up for this. So yeah, thank you so much again for coming to speak with us. Exactly, more Paul for us. Thank you again for coming to speak with us. It was a wonderful talk. I can stop recording at this time.